Welcome everybody to today's live webinar on everyday cell culture practice, improve reproducibility of your cell culture. And we're sending live from beautiful Hamburg in Germany. Our webinar will be presented to you by my lovely colleague, Dr. Jessica Wagener, application specialist for cell handling here at Eppendorf. And I will give you a short introduction about her in a minute. My name is Christian Haberland, marketing manager for instruments and systems here at Eppendorf, and I will be your moderator for today. At the end of Jessica's presentation, before we will start the live Q&A session, I would like to give you a quick overview about some of our solutions that might help you to improve your daily cell culture work. And before we begin, I would like to encourage you all to get the most out of this webinar by submitting your questions to Jessica at any time you want during the presentation. Just simply type them into the Ask a Question box on the left and click Send. And we will try to answer as many questions as we have time for at the end of the webinar. If you have trouble to see or hear the presentation, click on the Support tab at the top right or report your problem by clicking on the Ask a Question box on the left. And if you're interested in continuing education credits, just click on the tab at the top right. Now, let me shortly introduce the presenter of the webinar today. Dr. Jessica Wagener holds a PhD in biology with a focus on and many years of experience in cell culture. And she is one of our most experienced cell handling application specialists here at Eppendorf. Pre-COVID, she shared her knowledge in workshops and seminars around the world, and we all hope that the global situation will allow her to continue that soon. And you can find a complete biography of Jessica by simply clicking on the Biography tab at the top. So today we are happy that Jessica is going to share her knowledge with us. So I will start talking now. Let's get started. Jessica, the stage is yours.
Thank you, Christian, for the nice introduction. Welcome also from my side. Welcome to this today's presentation, and thank you for joining our webinar. After a short introduction, I will talk about some general aspects that influence reproducibility of different types of cell culture experiments. We will then have a look at the routine culture steps and outline the best methods to improve overall cell quality. In the end, I will touch the topic of misidentified cell lines and cell authentication. I want to start with showing you some results of a survey conducted by Nature magazine. In this survey, more than 1,500 researchers of all different scientific disciplines have been asked if they see a current crisis in reproducibility. And as you see here, more than 50% of researchers surveyed agreed that there is a significant crisis, and taken together, around 90% do see reproducibility as a problem in science. Shown here on the right-hand side are the responses to the question if there are any measures undertaken to enhance reproducibility. And although the topic seems to be on people's minds when you have a look at the results of the first question here on the left, still more than 1/3 of the researchers have never established any procedures for reproducibility in their lab. No matter which scientific field you look at, there are a few general principles that should be followed in every scientific process. A rigorous experimental design, including proper statistics for robust and unbiased results. A clear and complete documentation of both the experimental procedures and the obtained results. Transparency in reporting results and experimental details, and the reproduction of results are cornerstones of good scientific practice. Using cultured cells as model system has indisputable benefits. Especially primary cells retain many functions and properties of their parental tissue, which makes them an ideal model system to test new therapeutic approaches. From initial target validation studies through clinical candidate selection to subsequent translational studies, the use of continuous cell lines as a renewable resource of cell material is essential across numerous areas of biomedical research. But the cultivation of eukaryotic cells has some major drawbacks that, when overlooked, can lead to severe problems in reproducing experimental data. I think everyone working in a cell culture lab is aware of the risk of having contaminations with microorganisms that overgrow and ruin the cultures. Here, especially mycoplasmas are difficult to detect and are therefore a special threat in cell culture labs worldwide. Cross-contaminations of different cell lines are a widespread problem that endangers the reliability and reproducibility of experimental data. Besides microbial and cross-contamination, it is often overlooked that continuous cell lines, which have an unlimited lifespan, can undergo genetical changes. So, although the cells continue to proliferate, their genotypes and phenotypes can change over time. The cultivation conditions can have great effects on the cells, and here we are not only talking about specific experimental conditions that result in different cellular responses. It is about media formulation, the use of serum, or such simple things as incubator door openings. Cells can respond to the smallest influence. All these risks and drawbacks need to be managed for cell culture experiments to be reproducible. And with this webinar, I want to give you some practical tips how to incorporate certain standards into your cell culture routine that help to improve reproducibility. But before we proceed, here comes our first poll. As you will see, it is a very general question, and I'm curious about your answers. So, do you think that cell culture experiments are more difficult to reproduce than others? And the answers you can click on are yes, no, or I don't know. So, what do you think? Are cell culture experiments more difficult to reproduce than others? Yes, no, or I don't know. And I will wait a couple of seconds and give you time to vote, and then I will share the results with you. So I can see that the numbers are slowly rising and coming in. Okay, I will wait a couple of seconds more. Okay, so the answers are pretty clear. We have around 60%, 57% of you says yes, cell culture experiments are more difficult to reproduce. 26% says no, and 7% is not sure about it. So thank you for participating. So let's have a look at the steps that are usually part of the cell culture workflow independent of the specific cell type you are working with. And I want to start with the two steps here on the right, the seeding of cells that are used for an experiment. So this is what we are looking at in the next chapter, how to ensure best experimental conditions. Okay, so let's have a closer look at this first experimental step that is so important, the seeding of cells. The precondition for getting comparable results is to have comparable numbers of cells for every negative control, positive control, and for every experimental replicate. Variations in seeding cell numbers and the formation of air bubbles during pipetting will not only increase standard deviations, it will also make your results less reliable. When you fill, for instance, a multi-well plate with cells, it will take some time until you have filled all the wells. You can use a single-channel pipette and have the cell suspension in a tube, or you can use a multi-channel pipette and have the cell suspension in some type of reservoir. No matter which tool you are using, the longer the seeding process, the higher is the variation in cell number from one well to the other. As you see in the image here on the upper right, the cells sink towards the bottom of the tube with time, and this sedimentation goes quite fast, within minutes. Therefore, you need to resuspend or mix the cells to swirl them up in between to get an equal number of cells in every well. In saying that, we should keep in mind that cell culture media tend to foam due to the high protein content of supplemented serum. Air bubbles can hinder cell attachment, and in the lower image shown here, you can see how an air bubble hinders the cells nearby to adhere properly to the surface. So you should avoid harsh and fast pipetting to reduce the formation of air bubbles, and also to reduce shear forces which further stress the cells. Okay, here you see how a classical micropipette works. It is a so-called air cushion system. By pushing the control button, you push down the air above the liquid, and the liquid gets released. With this system, you have variable influencing factors, which I will talk about in the next few slides. They are best suitable for aqueous solutions, and if you do not work with filter tips, the inside of the pipette can be contaminated by aerosols. Positive displacement system, which you probably know as dispenser or stepper, function without air cushion. The piston is part of the pipette tip itself, and it's in direct contact with the liquid that is pushed down and released. So these pipetting systems are contamination-free, and they are unaffected by the type of liquid you are pipetting. And by the way, as there is no air inside the dispenser tip, you cannot create any air bubbles during pipetting. I already talked about pipetting different liquids. So when you treat cells with any recombinant proteins or inhibitors, these are often supplied as lyophilized form and need to be dissolved before use. Commonly used dissolvents are alcoholic solutions like ethanol, as well as DMSO. Both liquids can be difficult to pipette. Ethanol is a volatile liquid that tends to drip out of the pipette tip. With DMSO, you have a viscous liquid. So what some people do is they cut the tip of the pipette tip to create a wider opening. In both cases, alcoholic solution that is dripping out of the pipette tip and a tip that is cut to create a wider opening for viscous liquid, you cannot pipette an exact volume. But especially when pipetting any critical proteins, drugs, or inhibitors that you want to test with your cells, exact volume is essential. Just imagine you want to test different concentrations of a certain substance and you cannot be sure if the volume you pipette is precise. So looking at the different pipetting systems I talked about in the previous slide, a dispenser is the better choice here because its precision is unaffected by the type of liquid you are pipetting. Not only the type of liquid influences the pipetting results, also the pipetting technique plays a role, especially when using air cushion pipettes. Hold the pipette vertical when aspirating the liquid, as seen here on the left image. Immerse the pipette tip only slightly and try to keep a constant immersion depth. If you follow this technique, you will improve the precision of the volume that you are pipetting. And how the handling of cells, and to be more precise, how pipetting of cells can influence your experimental outcome is shown on this slide here. In this graph here, you see the results of a cell viability assay. Each column represents one row of a 96-well plate. 2 rows were left out for blanks. The aim was to seed an identical number of cells in each well. And the cells were plated with a manual 8-channel pipette, and after 24 hours, the number of viable cells was determined using a colorimetric assay. In rows 1 to 5, you see varying cell numbers in the wells and therefore high standard deviations. Here, the user did not mix the cell suspension between pipetting each row, so the cells sedimented in the reservoir with time. In addition, the pipette was held in different angles and heights during aspiration. In row 6 to 10, you see the results with mixing the remaining cells before pipetting each row, aspirating at constant height and in upright position. Taken together, these data show that the pipetting technique can have a big influence on the results of a cell-based assay. Besides seeding equal cell numbers, when we are talking about adherent cells that attach to the vessel, you also want to have a homogeneous distribution of the cells on the growth surface of that vessel. On the lower left image, you see what happens if you pre-fill the vessel with medium, add the cells, and that's it. The cells that were fixed on the surface and stained with crystal violet in this case mainly adhere in the middle where you have brought them in with a pipette. There are different techniques how you can disperse the cells evenly. Some people use a figure-8-like movement, others prefer a cross-like movement of the plate or the dish. As you can see here, both techniques lead to a better distribution of the cells, but the smaller the diameter of the vessel, the less liquid movement you have. So instead of pre-filling the vessel with medium and add the cells in a second pipetting step, you can first dilute the cell suspension to the desired concentration in a tube, so preparing a kind of master mix that you know from a PCR, for example, and then add the complete volume in one pipetting step to the well. Thus, you have an even cell distribution throughout the complete vessel and the cell attached more homogeneously to the growth surface. And here I show you an example how uniform cell adhesion can have an impact on your experiment. In these experiments, we transfected cells with a GFP expression construct and imaged them later on. As you see in these images, the transfection efficiency is sensitive to cell density. The optimum for transfection is a medium cell density. When you have an evenly distributed cell layer, you have a uniform transfection efficiency. And as you can see in this quantification, in the areas with medium density, you have the highest transfection efficiency, In this experiment, it was between 50 and 60%. In the dish with the uneven distributed cells, the efficiency varied between 26 and 56%, which in total gives you a lower transfection success rate and a much higher standard deviation. Let's come to another factor that can influence cell attachment and growth: vibration. Here are two simple examples. On the left, you see adherent cells that are exposed to frequent door openings in the first few hours after plating. These vibrations can cause uneven adhesion of cells to the substrate they are growing on. On the left, it is shown what happens when you move the vessels. No, I'm sorry. On the right is shown what happens when you move the vessels with freshly seeded cells. So, for example, you want to put your Petri dishes into a crowded incubator and need to push other vessels aside to make space on the shelf. This manual moving can disturb even cell adhesion and distribution. External vibration sources in the lab include running centrifuges, fridges, or air conditioning systems. Some incubators have a fan inside, which is obviously one of the most critical sources of vibration because the cells are placed more or less directly next to the fan. So whenever possible, incubators without internal fan should be preferred, especially for sensitive cells or sensitive experiments. The conditions for the cells in the CO2 incubator should be as stable as possible, of course. Every time the door of the incubator is opened, the atmosphere inside is disturbed. It is a good advice to reduce the traffic in and out of the incubator as much as possible. Only open the door when necessary and close it as soon as possible. The incubator should have a short recovery of CO2 and temperature levels after door opening. Some incubators achieve that by an overshoot, especially in temperature, but this can be harmful for the cells. So a good incubator is able to recover temperature and gas level within a few minutes after door opening and without any overshoot. Especially when you work with very sensitive cells or when you work in a lab where many people are sharing an incubator, it can be a good idea to have separate incubators for maintenance cultures and running experiments. So you seed the cells for an experiment, do the treatment, manipulation, or whatever you want to do, and put the cells into a separate incubator for the time of the experiment. Your colleague who must split the cells or change the medium uses the other incubator, and thus your experiment is not disturbed by fluctuations of the incubation atmosphere. Depending on the organization and work routines in your lab, there are different ways to organize the contents in your incubator to easily relocate cells and further reduce number and duration of door openings. Shown here are a few different examples. Incubators with small segmented inner doors that can be opened separately are a good measure to keep the disturbance of the incubation atmosphere to a minimum. In addition, opening a small segmented door instead of a big single door speeds up recovery as shown here in this graph. And it also reduces consumption of CO2 gas, which reduces cost and labor for exchanging gas bottles, and it improves the carbon footprint of your lab. So again, here's an overview of the general cell culture workflow. In the next chapter, I want to talk about some aspects, how you can improve the overall quality of your cells, because not only the experiment itself plays a role when it comes to reproducibility, also the routine steps like freezing, passaging, and the cultivation itself can be a critical factor. Okay, but before we go on, I would like to start the next poll question. I'm interested in the cell types you are working with. The answers you can click on are continuous cell lines, primary cells, or both. So again, I will wait a couple of seconds and give you time to answer. So what cell types are you working with? Continuous cell lines, primary cells, or both? And again, we will wait a few seconds. I can see the votes are slowly coming in right now. Okay, so far it looks like the majority of you is working with continuous cell lines. I will wait a couple of seconds more. Okay, here we go. So we have 45% is working with continuous cell line, only 20% is using primary cells, and 36% is using both continuous cell lines as well as primary cells. All right, so thank you for participation. All right, so let's have a look at the next chapter. I want to start with the cryopreservation of cells and cell banking. During freezing and thawing of cells, the stress factors should be minimized. Having high-quality cell stocks is your backup system and the prerequisite for reliable research. Maybe the most important point is that you make sure that you don't freeze contaminated cells and that the cell line is really the cell line you think it is. So authentication of cell lines should be performed regularly, especially when you got the cells from another lab and when you use them in long-term or subsequent studies. I will come back to that topic in a few minutes. The cells you are working with should also be tested for mycoplasma regularly. The routine use of antibiotics is not recommended because they can mask a contamination with partially resistant bacteria. And by the way, most antibiotics are not efficient against mycoplasma anyway. Filter tips give extra safety, so they should be considered at least for such important steps like cell banking. A two-stage cell banking system with a master and a working stock, like shown here on the right, provides you constantly with early passages from your cell lines. And finally, a detailed record of the cell stocks is as important as clear labeling of cryotubes. No matter what you do with a cell culture, freezing, subculturing, or plating cells for an experiment, it is important to determine the right time point for a particular cell type. In this image, you see a typical growth curve of cultivated cells. It is important to not let the cells overgrow until they are in the stationary phase and do not proliferate anymore. So the optimal time point is when the cells are in their exponential growth phase, also called log phase, and this is when they are between 70 and 80% confluent. That would be the perfect time for splitting them. Plotting a cell growth curve and determining the population doubling time for a cell line is critical for understanding the optimal seeding density and the optimal cell concentration range for the routine subculturing of that cell line. When you launch an experiment, it is important that you can be sure that any changes in cellular behavior are a direct response to an experimental stimulus. For example, you want to test how the cells react to the treatment with a certain substance. So you add the substance to your cells and see increased cell death or the proliferation rate slows down. To evaluate a direct correlation between these changes in your cell culture and the experiment you performed, a constant culture routine is important. A growth curve provides information about cell behavior during cultivation and thus facilitates the assessment whether any biological response can be attributed to the effect of a tested factor or whether it may reflect an imbalance within the culture system. Another factor that has often been described to influence cellular behavior is the passage number. The passage number is a record of the number of times a cell culture has been subcultivated. There are numerous studies giving evidence that the passage number affects a cell line's characteristics over time. It has been described that cell lines at high passage numbers experience alterations in morphology, response to stimuli, growth rates, protein expression, transfection efficiency, and more. So, looking at all these passage number effects in different cell types described in the literature, the basic question one can think of is how many passages are too many? And before we go on, I would like to hand over this question to you and start our next poll. What do you think? How many passages can a cell line be cultured for? And the answers you can click on are not more than 20 passages, 20 to 60 passages is okay, up to 100 passages, or the passage number has no influence on the cells. So what do you think? How many passages can a cell line be cultured for? Again, we will wait a few seconds and then we will share the results. So the votes are slowly coming in right now. There is already a clear tendency to the first option, not more than 20 passages, but I will wait a couple of seconds more because there are still answers coming in. Okay, I think we can close the poll now. All right. Okay. So around 53% of you says not more than 20 passages is okay. 35% is using their cells between 20 to 60 passages. And only 10% is using the cells up to 100 passages, and then there is only 2% of you who says the passage number has no influence on the cells. All right, thank you for participating. So in general, we can say keep the passage number as low as possible. Especially primary cells that have a finite lifespan are prone to genotypic and phenotypic changes as they adapt in vitro culture. After a number of population doublings, they will simply stop growing. In contrast to that, continuous cell lines like cancer cell lines have an unlimited lifespan. But although they can proliferate indefinitely, also their genotype and phenotype can change over time, as I already mentioned in the beginning of the webinar. A lot of the well-established and well-characterized cell lines that are used in labs worldwide have been isolated from their original tissue decades ago. So when you work with such an old cell line, it is most likely not possible to obtain early passages from the cell line. But you must set a starting point as a reference to be able to obtain reliable results with these cells. First of all, the starting point should be cells of high quality. The best way to ensure this high quality is obtaining cells from one of the established cell banks. Here you get well-documented, authenticated cell lines, which serve as the best reference cultures for your experiments. By establishing a two-stage cell banking system, like I showed you in one of the previous slides, with a master and a working stock, you have a constant supply of early passages from your cell lines. Now you can determine the safe passage number for your cell type of interest. By establishing certain baselines, for example, test the cells for the presence and expression level of a certain protein. You can monitor the proliferation rate over time. Keep an eye on any changes in cellular morphology. By getting to know your cells, you will get an idea of whether and how the cells change in relation to prolonged passaging. And maybe a very simple tip, do not keep cells on culture that are not actively used for experiments. So don't passage continuously, but rather establish new cells from frozen stock if required. As I already mentioned, it is important that you get to know your cells, no matter which step you perform. First, take a closer look with the microscope. Not only check the cells for possible contaminations, but also to reveal the status of your cultures. There is a lot of things besides contamination and confluency that can be checked with a simple bright field microscope. We already heard about techniques to avoid that the cells accumulate in one area of the growth surface, like you see in this image in the upper right compared to the lower left. Other things that should draw your attention when looking through the microscope are changes in cell morphology. So, for example, increased granularity, especially those observed around the nucleus, or vacuolation of the cytoplasm can be indicators of an unhealthy culture. In some cases, it simply requires a change of medium, whereas in other cases, a more serious problem may be present, such as microbial contamination, senescence of the cell line, or inadequate medium or serum. As I already mentioned, cell culture media tend to build up a lot of foam, and air bubbles easily form during pipetting step. Especially when you work with very small diameter vessels, the air bubbles can hinder cell attachment when seeding the cells, as we already heard a couple of minutes ago. When cells die, they will take on a round appearance, like you see here, detach from the surface and float in the medium. Increased cell loss due to apoptosis or necrosis is a clear sign of an unhealthy culture that should be replaced by starting a fresh culture from frozen stock. As thawing is always a stress factor for the cells, it is normal to observe a number of dead cells floating in the medium of a recently thawed culture. A medium change should be performed one day after thawing the cells to avoid accumulation of toxic products from dead cells in the medium. Proliferating cells are a sign of a healthy culture. When cells undergo mitosis, they assume a round shape and partly detach from the surface before they segregate into two daughter cells that will adhere to the surface and assume their characteristic morphology again. As mentioned before, when cell density is too high, cells will stop proliferating. For this reason, passaging at 70 to 80% confluency is recommended. Constantly monitoring of the cells is crucial to reveal possible contamination at early stages and avoid spreading of the contamination. For microbial contaminations like fungi, yeast, and bacteria, a standard bright field microscope is sufficient to detect them. In a lot of cases, they are even visible with the naked eye as color changes and turbidity of the medium appears. There is one type of bacteria that is not so easy to detect, though. When a contamination with mycoplasma or any other type of contamination goes undetected, it endangers the reliability and reproducibility of experimental data. Mycoplasma are too small to be seen in a standard microscope, and they don't visibly change the culture medium. The risk of spreading a mycoplasma contamination throughout the lab is high. Therefore, cultures need to be tested regularly. In this graphic here on the right, you get an idea of the dramatic intracellular effects caused by a mycoplasma infection. There are not many cell culture quality standards defined in detail and are thus performed differently between laboratories and sometimes even within the same lab. Usually everyone is treating their cells a bit different, and this is one of the reasons that contributes to non-reproducibility of experimental data. A constant and detailed documentation helps to ensure that a cell type is always handled the same way. The source should be recorded by quoting the original designation of the cell line. The accession number should be retained if the cell line was obtained from a cell bank. Especially when establishing a new cell line, a unique name prevents ambiguity with respect to other cell lines or biological resources. Also important is an overview of culture conditions and procedures, including information about medium composition, growth surface, thawing density, etc. The culture conditions should be adapted for each individual cell type. Reference images of the cells at different densities help to recognize any unusual changes in cell morphology. Especially when new people come to the lab or when you start to work with a new cell line, these images help to get familiar with how the cells are supposed to look like when the culture is healthy. Images with different cell densities show how the morphology can change with increasing confluency. In cell culture labs, it is common that different people take care about the cells during the week. One day, person A is changing the medium, the next day, person B is splitting the cells. But even if you are the only person working with the cells, things like passage number, seeding number, split ratio, as well as cell viability should be constantly documented. So taken together, implementation of standardized laboratory practice protocols contributes to the establishment of comparable parameters for high reproducibility, independent of the person performing the experiments. When we talk about documentation and monitoring to improve reproducibility, the CO2 incubator should be part of it. Here on the lower right, you see a screenshot of the menu of the Eppendorf Cell Expert CO2 incubator. The red arrow indicates a door opening, and you see that the curves of CO2 and temperature are dropping. Monitoring cultivation parameters and documenting performance of your incubator are measures to establish long-term reproducibility of experiments in your lab. You can simply check for door openings or track back the performance data to see what might have influenced your experiment this time. So looking into the performance of your incubator should be part of a troubleshooting of an experiment. There's one important source of variability that cannot be overlooked when we are talking about reproducibility in cell culture, and that is animal serum. It is a standard supplement of cell culture media. The most commonly used serum type is fetal bovine serum, also known as fetal calf serum or FCS. It is added to the culture medium at a concentration of 10 to 20%, and it contains vital nutrients, hormones, growth factors, which stimulate cell growth. Other serum components serve as binding proteins, which promote cell adhesion in vitro. Although the use of serum is well established in cell culture, it remains the most undefined component of cell culture media. Serum batches usually show qualitative variations, and the lack of uniformity in composition introduces high batch-to-batch variability. The development of serum-free media and serum substitutes is increasing. A variety of chemically defined serum-free media and synthetic serum substitute is available from different suppliers. Despite of the ethical issue that come along with the use of animal-derived substances, for some applications it is crucial to have a complete animal-free culture conditions. One example are bioprocess applications, such as the production of therapeutic proteins or vaccines. Also for the cultivation of stem cells, researchers want to resign the use of serum because stem cells are known to react very sensitive upon the slightest variation in culture conditions. However, there is not yet an alternative for all cell types available. In addition, the alternatives to FPS that are available are usually very costly. So there are a few things you might consider to avoid the variability of serum affecting cell-based experiments. It is recommended to test different serum batches for their ability to support growth of a certain cell type and to stock up on a suitable batch. It is also possible to request that serum suppliers reserve appropriate volumes of a suitable batch, so when the batch is used up, testing should be repeated to identify the next suitable one. I want to point out here that when you test a new batch of serum and find it to increase the growth of your cell significantly, it is not necessarily beneficial for the reproducibility of previous results you obtained with these cells. You should not simply screen for the lot or the batch that make the cells grow fastest. So, as I already mentioned, for finding a safe passage number for your intended study, testing of different serum batches should be tailored for your intended use. So, for example, have a look at the expression of certain marker genes of interest and monitor cell morphology with different serum batches. The last important point here is transparency. When you report how you screen or test serum, you enable others to reproduce the data more easily. Okay, and with this I come to the last chapter of this webinar, misidentified cell lines. So a misidentified cell line develops from a cross-contamination of two different cell lines. Usually this happens by an accident somewhere during the handling of cells. Mislabeling of samples and inconsistent nomenclature of cell lines also contribute to this phenomenon. As soon as two cell types are mixed up, it is very hard to distinguish those two. This process goes fast and it might go completely undetected, especially if the morphology of the cells is not drastically different. But although the phenomenon of cross-contamination and misidentification of cell lines has been recognized for more than half a century, still to date we know that between 15 to 20% of all cell lines used are either cross-contaminated or misidentified. Currently, more than 500 cell lines are listed to be cross-contaminated or misidentified. Misidentified or cross-contaminated cell lines are a big contributor to non-reproducible research. Therefore, it is very important to check the identity of the cell lines you are working with. Identity testing can be performed by using short tandem repeat profiling or STR profiling. The analysis of short tandem repeats has become the standard for intraspecies identity testing of human cell lines. Okay, and with this, I will hand over to Christian again, who will tell us something about Eppendorf cell culture products, and then we will start the Q&A session. Okay, so Christian, it's up to you.
Thank you very much, Jessica, for your presentation. So, as I promised you in the beginning, and before we will start the last poll question and the Q&A session, I promised to give you a little insight into helpful product solutions for your daily work. And the first thing we will... Oh, there is the poll. So, then we do this before. And the question is, how do you rate the content of this webinar? Very good, good, okay, or poor? I'll give you a few seconds for this, or we go over to the next question, or the next slide. So here is the little product solution part. So the first thing I would like to introduce to you is the Cell Experts Util Incubator family. Here are some details about this. So as we mentioned before, it can be harmful for your cells if you're using incubators with a fan. That might be one of the problems in your cell culture. So we at Eppendorf strongly believe in incubators without fans, so they are very easy to clean. They also come with high temperature disinfection and more. They are very reliable when it comes to contamination prevention. On the other hand, in the past, it was always the case that incubators with a fan recovered atmosphere faster than fanless incubator, but actually this is not true today anymore. So the cell export is actually, so it's true that no other incubator in the market recovers temperature and CO2 faster than the cell export here. It also helps you to save some money. So up to 8,300 euro or $9,000 over 5 years by significantly reduced gas consumption, for example, and it also provides up to 25% more usable space. And if you're interested in this incubator, go to eppendorf.com/Q2-incubators. The next product I would like to introduce to you is our new SmartBlock cryo thaw for standardized water-free cryo vial thawing, because we know that a lot of cell culture customers are thawing their cryo vials either in the hand, which is not good if you rely on standardized processes, or in the water bath, which poses a high contamination risk. So this is actually a workaround, especially if you already have a Thermomixer C or a thermostat C from Eppendorf in your lab. You can just simply use the SmartBlock for simultaneous and reproducible thawing of up to 24 cryo vials, and we have also tested this for very sensitive cells like human iPSCs. And if you're interested in that, go to eppendorf.com/mixers. The next product, and during my lab time, I would have appreciated to know what this is. It's called Multipette or repeater, depending on the region you're in. And it's actually a positive displacement pipette or dispenser. Jessica said something about this in the beginning. And what you can do with this is to aspirate once and pipette up to 100 times. And regardless of the liquid type, you're always correcting the correct volume. So regardless if you're doing or working with DMSO, ethanol, or serum, you always have the correct volume without dripping out of the pipette tip. And it also, of course, helps you to have a high contamination protection because all the single tips are sealed against the inside of the pipette. And you can also, because it's an electronic system, use the speed controls for more safety for your cells, so you don't have shear forces for very sensitive cells. And also with the system here, you have an automatic tip recognition, so you can save time for any type of volume calculation, so that the multipette is directly, or the repeater, recognizing the volume of the tip that's attached to it, and you can do or save time for your volume calculations. If you're interested in that, go to eppendorf.com/E3. So, and with this, we will come to our live Q&A session. So if you have any questions you would like to ask Jessica, please do so now. Just click on the Ask a Question box located on the far right of your screen, far left of your screen, and she will answer as many questions as we have time for. And I've already seen that there are a lot of questions during the presentation. So we will start right away with this last question, or with this first question. And the first question is, when growing cells in suspension, both to separate viable and non-viable cells for passaging or freezing?
Yes, thank you, Christian. So basically it is the same than you do with adherent cells. So of course, when you have adherent cells, you instantly see the dead cells because they will float around in the medium, of course. But you can, for suspension cells, you can do a very simple trypan blue staining, so you can distinguish dead cells from live cells, and it's a very simple protocol. Just add the trypan blue and wait a few minutes, and then you see or you can distinguish dead from live cells.
All right, thank you very much. The next question that we have in our chat here comes from Kanmani from the National Institute of Technology in Durgapur. And the question is, does the passage number depend on Hayflick's limit?
No. So the Hayflick's limit is the limit of population doublings of a certain cell type. So when you have a primary cell that is freshly isolated from the tissue, it can undergo around 60 cell division before it starts senescence and dies. So, and that is the Hayflick's limit. The passage number is more or less an artificial number that you give to the cell line when you passage it. So it has nothing to do with the Hayflick limit. I hope that answers the question.
Yeah. So let's move on to the next one. And the next question comes from Noor from India. And the question is, will it be possible to get rid of Mycoplasma 100% from cell cultures with available Mycoplasma removal kits?
I would love to say yes, it is possible. But it always depends on the status of your Mycoplasma infection. So how many Mycoplasmas do you already have in your culture? What Mycoplasma species do you have in that culture? So if you have, of course, there are cell types that are irreplaceable. So you can try to get rid of the Mycoplasma by using such a Mycoplasma removal kit, or also using subsequent different Mycoplasma kits, but there's no promise that it will be successful to 100%, and B, that it won't have any influence on the cells you are culturing. So you should always be aware that it might also have an effect on your cultured cells. So if you can avoid using these Mycoplasma kits, I would recommend to simply discard infected cultures and start with new cultures from frozen stock. But you can try, but there's no guarantee that it will be successful 100%, unfortunately.
Thank you, Jessica. Here's another interesting question that's related to Mycoplasma contamination. And the question comes from Volker. And the question is, contamination of stocks with Mycoplasma in the liquid nitrogen tank, is that a real danger or just a fairy tale?
The question is a classic. I get this question very often. I have to say that I actually don't know, but I heard it so many times now that I start to believe it. What we know is that cryotubes are often not 100% tight, or you cannot close them 100% tight. So, and when you have placed the cryotubes directly in the liquid phase of the nitrogen, there is, of course, a chance that liquid nitrogen goes into the tubes and goes out of the tubes. So I could imagine that you can transfer Mycoplasma from one tube to the other in a liquid nitrogen tank when you have the tubes in the liquid phase of the nitrogen. But I have no evidence for that. So we never tested that. But I heard it from many people. I heard a lot of stories about the whole liquid nitrogen tank that is infected with Mycoplasma. And yeah, I tend to believe it, although I cannot prove that it is right or that it's wrong.
So I guess Mycoplasma is still a very big topic because here's also another question, and that is, what is the best method to detect Mycoplasma?
There are a few different methods available. You can go for ELISA or any biochemical assays or PCR. PCR is a very straightforward method. There are a lot of commercial kits available covering the most frequent Mycoplasma strains that are affecting cell culture laboratories. I would not recommend to rely on a simple DNA staining like a DAPI staining or a Höchst staining, because here you cannot distinguish if if it's the DNA from the mycoplasma that are stained, or if it's the DNA from any cell debris that is stained. So such a DNA staining should only be a first step. If you think that could be a positive result, I would go for a second detection method like PCR.
Thank you very much. So I have to choose wisely amongst the many questions here. So I will pick this one here. And the question is, does the use of antibiotics in cell culture media influence reproducibility? Always a good question.
Sorry, could you say that again? I didn't hear that.
Yes, I do. The question is, does the use of antibiotics in cell culture media influence reproducibility? Is there any evidence for that?
There is quite a lot of literature out there that shows that antibiotics have a certain effect on the cultured cells. And the thing is that when you have literature of a certain cell type, you never know if this piece of literature or if the cells were cultured with or without antibiotics, because this is usually not reported in the material and methods section. No one reports which antibiotics they are using, how much they are using. There are people that add fresh antibiotics every time to the medium. Others only add it once, and then they continuously preheat the medium until the complete bottle is empty, and antibiotics are quite temperature sensitive. So yes, I would say antibiotics can have an influence, and I would always try to avoid the routine use of antibiotics because it also gives you kind of a false sense of security, because you will just increase resistant bacterial strains, and when you have a partially resistant contamination, it's at a very low level. It is very hard to detect, and by adding antibiotics, you do not necessarily kill them. You just make sure that you still cannot detect them, because it does not pop up like it would without antibiotics. So I would not recommend a routine use of antibiotics.
So it's not the magic bullet.
Exactly.
All right. Let's see if we have a question coming from a different direction. So maybe that one is interesting. The question is, should I centrifuge the cells right after thawing them?
Yes and no. That can be. It is dependent on the cell type you're working with. So there are cells that react very sensitive to the centrifugation step right after thawing. Other cells react more sensitive to DMSO when they are thawed. So if you are not sure which would be the best procedure for your cells, I would simply do a parallel test with freshly frozen cells. One vial is centrifuged right after thawing, the other one is not. And then you can compare the viability of these cultures like 48 hours, 24 hours after it. So no matter if you centrifuge or if you don't centrifuge, I would definitely change the medium one day after thawing to get rid of any cell debris or of any DMSO that might be in the medium. But there is no ultimate answer here. It will depend on the cell line.
Thank you very much. So I guess we have time for maybe one more question. So I have to choose wisely, and as there are so many questions on mycoplasma, I choose this last one on mycoplasma, and the question is, how often should cells be tested for mycoplasma contamination?
Also a very common question. It depends on how your lab is organized. So are you working in a very small lab with only a few people who are very experienced in cell culture or in aseptic techniques? You can choose a lower frequency of testing when you work with many people sharing equipment, sharing incubators, consumables, reagents, and whatsoever. And maybe also when you have a lot of not so experienced people in the cell culture, I would choose a higher frequency. So I would always start with a high frequency of testing, let's say every 4 weeks, and then have a look at the results. And if you have subsequent negative tests, you can think about lowering the testing frequency maybe to every 6 to 8 weeks or something like that.
Thank you, Jessica. Do you have any final comments for our audience today?
Thank you for your attention, and thank you for joining our webinar today. And thank you for all the interesting questions that were coming in. There are a lot of more questions I can see in the chat, but we do not have more time. So I will come back to you via email and try to answer all of your questions. So give me a few days time. And then you will get an email with the answer to your question. Thank you for your attention again.
So, and before we go, I would like to thank you all for joining us today and for sharing your questions. As Jessica said, questions that we did not have time for today and those were submitted during the on-demand period will be addressed by Jessica via the contact information that you've provided at the registration. I would like to thank Jessica for her time today and for a brilliant presentation. And we would also like to thank Michelle Ashton and the whole LabRoots team for supporting us today. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. So feel free to share it, share the recording with your colleagues. And also, if you're interested, check out our next webinar that is together with our partner Promega on cell line identity that you can see here on the screen. It's on preventing cell misidentification in your daily lab routine on June 9th here at LabRoots. So we say goodbye from Hamburg. Stay happy and stay healthy.